Hi, everybody, and thank you for joining us. My name is Suzanne LeBlanc, the Director of Clinical Relationships at the Focused Ultrasound Foundation. I am proud to partner this webinar along with Yanni Wattenmaker, the Executive Director of Breast Cancer Alliance. In honor of Breast Cancer Awareness Month, we are thrilled to present this webinar for new approaches with immunotherapies for breast cancer, as both organizations support research exploring the potential of harnessing our own immune system and controlling the tumor microenvironment to fight breast cancer. Although advances in diagnostic imaging and increased public awareness has improved earlier detection, the medical community continues to develop novel strategies to decrease the need for invasive surgical procedures, minimize the side effects from radiation therapy, improve the targeted delivery of chemotherapies, and importantly, harness the power of our own immune system to improve clinical outcomes. The presentations you will hear today discuss this overlap of the FUS Foundation research and Breast Cancer Alliance sponsored research. On the focused ultrasound side, there are about 11 clinical trials using 6 different focused ultrasound machines for patients with early and late stage breast cancer. Yanni, would you like to say a few words?
Sure, and thank you so much for having me today. It's a pleasure to represent Breast Cancer Alliance and see some of our grant recipients on this panel. So Breast Cancer Alliance works diligently all year long to fund not only early stage research, but also the training of future breast surgeons and to support the needs of underserved patients, getting them the access to care that they so desperately need. But what we particularly enjoy about our place in the research field is this niche sort of space of venture capitalism, if you will, or bootstrap capital. So what we like to do is we like to come in and fund very innovative hypotheses and give the researchers the opportunity to showcase what's possible when others are still skeptical. And we have a wonderful scientific review committee who allows us to do that. And Doctors Nemeth and Abrams are beneficiaries of that funding, doing extraordinary work of which we are so proud. So we're really grateful for the opportunity to help to showcase that work today and give the viewers who don't know about the work of Breast Cancer Alliance an opportunity to learn a little bit about what we do and see the impact of our funding. So thank you.
Thanks, Yanni. That's a great background information about your organization, which is fantastic. And I've enjoyed getting to know you and your organization over the past 2 years. Now let's introduce our panelists. First, we will hear from Dr. David Brenin, a professor in the Department of Surgical Oncology at UVA and Division Chief of Breast and Melanoma Surgery. He is also co-director of the UVA Focused Ultrasound Cancer Immunotherapy Center, and he'll discuss the evolution of focused ultrasound treatments for breast tumors that started with benign fibroadenomas and then progressed to advanced and early stage breast cancer. Next, we will hear from 2 researchers at the Roswell Park Comprehensive Care Center. Dr. Scott Abrams is a professor of oncology in the Department of Immunology and co-leader of the Tumor Immunology and Immunotherapy program. Dr. Michael Nemeth is an assistant professor of oncology in the Department of Immunology. Now, a special treat, following these presentations, Dr. Natasha Shabani will moderate the discussion. She's a newly appointed assistant professor of biomedical engineering and radiology and medical imaging at UVA, who just received the prestigious NIH Director's Early Independence Award. She's also the research director at the UVA Focused Ultrasound Cancer Immunotherapy Center. I'm so excited for you all to hear these presentations, so let's get started.
Good morning, everyone. My name is Dr. David Brenin. I am the Chief of the Breast and Melanoma Surgical Services at UVA, and this morning I'm going to give a brief update on the use of focused ultrasound ablation for the treatment of tumors of the breast at the University of Virginia. I have the following disclosure to make. Our initial experience at the FDA made it clear that the best approach would be to proceed in a stepwise fashion, first treating benign tumors of the breast with focused ultrasound ablation, then moving to treat advanced stage breast cancer, and then finally to treat patients with early stage disease. Moving forward with this stepwise approach, we began by treating fibroadenomas. Fibroadenomas are one of the more common benign tumors of the breast, but they can be quite bothersome to patients, producing a mass in the breast, often causing significant pain and tenderness, and if large enough, they can change the contour of the breast. The typical management of these tumors consists of observation or surgical excision. While fibroadenomas are benign, many patients are quite bothered by them and wish to have them removed. At this time, the most common option is surgery, but transcutaneous ablation has obvious advantages, and at the risk of being too obvious, I'm going to explain them here. This slide has two photographs on it. In both cases, the patient's fibroadenoma is gone. One patient underwent surgery, the other a transcutaneous ablation. Now, no matter how good a surgeon I am, in order to do an operation, I'm going to need to make an incision. And that incision is going to result in a scar. There's nothing I can do to avoid that. So again, in both cases, the patient is no longer able to feel her fibroadenoma. She is no longer bothered by it. But in one case, there's a scar, and the other, there is no scar. And that's the most obvious advantage to transcutaneous ablation, not only to treat benign tumors of the breast, but also to treat all tumors of the breast, including malignancies. Since focused ultrasound ablation is completely non-invasive, it requires no local anesthesia, it's fast. The entire patient treatment experience is typically less than 2 to 2.5 hours, and recovery is quick, usually back to work the day after surgery. We've used the EchoPulse device for all of our breast cancer studies, and it consists of a computer-driven system comprised of an electronics cabinet, an extracorporeal probe mounted on an arm and moved by motors, a cooling unit, and an ultrasound imaging probe that is mounted within the HIFU transducer. Here is the business end of the device. This is the roboticized head. This is a cooling jacket through which cool liquid circulates to keep the skin cold during the treatment. This is the ultrasound probe that is embedded within the HIFU transducer. IDE G130-252 was the first study we did at UVA evaluating the use of focused ultrasound ablation to treat tumors in the breast. It was a single-arm study in which we enrolled 20 patients. The primary objective was to evaluate the feasibility of ultrasound-guided focused ultrasound ablation for the treatment of breast fibroadenomas. We evaluated both tumor response and the patient's experience of the treatment. The primary endpoints for this study were palpability of the lesion at 12 months by both the treating physician and patient, how well the patient tolerated the treatment and their satisfaction with the treatment, and change in tumor volume as measured by physical exam and under ultrasound. We made the above measurements at 3, 6, and 12 months following treatment. So how is the procedure carried out? The patient is first positioned comfortably on a stretcher, and intravenous conscious sedation is provided throughout the procedure as needed. The mass is then localized with standard ultrasound imaging, and degassed local anesthetic is injected around the tumor. The transducer head is then positioned over the lesion to allow for optimal imaging of the mass, and targeting and treatment planning is performed in multiple planes throughout the lesion. This is what the treatment plan looks like on the device. Here is the fibroadenoma as viewed in cross-section, and here are the sonication zones individually depicted from the side. The treatment head then moves to the next cross-section, and you can see that here. Again, the device suggests multiple sonication zones to treat that cross-section, and these are viewed from the side here. In the middle, we're viewing those sonications from above, and what this is depicting is the entire treatment plan, where we paint sonication zones across the entire tumor in 3 dimensions, thus treating the whole lesion. This is a complete treatment plan as viewed from above, with sonication zones painted across an entire lesion. During the procedure, there are alternating ablation and cooling cycles. Each sonication pulse lasts several seconds, followed by an adequate cooling time, which typically varies between 10 and 25 seconds. The pulse power is first adjusted to create boiling within the tissue in the sonication zone, and then once we determine the adequate pulse power, the treatment progresses until all the sonication sites in the treatment plan have been delivered. So what does it look like? Well, on the device, prior to treatment, the fibroadenoma looks like this. A sonication pulse is then delivered, and this results in a hypoechoic mark as depicted here following the sonication. The treatment head then moves to the next sonication site, and another pulse is given. This heats up the second area in the treatment plan. The device then moves to the third area in the treatment plan. Another sonication pulse is given, and this continues till all of the sites in the treatment plan are sonicated. Following treatment, the lesion usually looks like this. So this is typically what we saw. These are images from our first patient. On the left, you have the pre-treatment ultrasound image with the volume for the fibroadenoma being 1.53 cubic centimeters. 3 months later, after treatment, the volume was down to 0.47 cubic centimeters. That's a 69% reduction in volume. And as it turns out, that was typical for the 20 patients that we treated. And as you can see here, there was a mean percent reduction in volume at 12 months for the 20 patients in that trial of 68.9%. Based on our results, we concluded that focused ultrasound ablation was feasible and effective to treat patients with fibroadenomas. It carried minimal toxicity and was very well tolerated, and we then moved on to opening up a multi-center, multinational trial to treat patients with fibroadenomas with focused ultrasound ablation, in which we'll enroll a total of 100 patients. That study is nearly completed with greater than 95% of patients enrolled, and we hope to conclude that study shortly. Following conclusion of the study, we will move to submit that data to the FDA, which will hopefully result in approval of this treatment in the United States. Okay, so we had some good experience treating benign tumors of the breast. Now it's time to move forward with treating breast cancers at UVA. So I wanted to leverage everything we had going for us at UVA, namely that we have a fantastic multidisciplinary breast cancer program. We have an internationally prominent immune therapy center headed up by Dr. Craig Slingloff, and we have our clinical focused ultrasound devices. I wanted to leverage all of this, so we decided we were going to move forward by treating breast cancer with a combination of focused ultrasound ablation and immune therapy at our program. Immune therapy leverages the patient's own immune system to help fight their cancer. An important component of immune therapy uses tumor-infiltrating lymphocytes. These are T cells, or a type of white blood cell, that are created by a patient's immune system and can help kill their cancer. In order to be able to do that, the tumor-infiltrating lymphocytes have to be able to traffic to or transport themselves to the tumor. Once they're at the tumor, they need to be able to infiltrate into the tumor and recognize the tumor as not self. The ability to recognize the tumor as not self, or an invader, is vital, because if the tumor-infiltrating lymphocytes are not able to do that, they will not attack the tumor cells and kill them. Unfortunately, breast cancer has lots of tricks up its sleeve to fool these tumor-infiltrating lymphocytes into thinking that the tumor is self. In other words, it's not an invader, it's not self. One of those tricks uses a protein called PD-L1. This protein normally tells a lymphocyte that a cell is self. Breast cancers can produce PD-L1 and convince tumor-infiltrating lymphocytes that it's not cancer, but part of the patient's normal tissue. When PD-L1 is detected by tumor-infiltrating lymphocytes, it tamps down the immune response. And for this reason, breast cancers that create PD-L1 fool the patient's immune system into thinking that it's not an invader and it should not kill the cancer. Additional ways that minimally immunogenic cancers can evade immune surveillance and confuse the patient's immune system into not being able to recognize a cancer and fight it include the ability to secrete type 1 interferon. This tamps down the immune response, similar to as I previously described. These tumors can also recruit what are called myeloid-derived suppressor cells and T regulatory cells. These are other types of white blood cells that again have the ability to confuse tumor-infiltrating lymphocytes and tamp down that immune response and have the tumor-infiltrating lymphocytes not fight the cancer. And then, as I mentioned previously, through the expression of programmed cell death ligand PD-1, which again downregulates the CD8 positive T cells' ability to fight the cancer. Here's a cartoon showing a very tricky breast cancer cell, and unfortunately this is commonly the case. That breast cancer cell is here, and it not only has an antigen for a tumor cell on its surface, but it also makes that antigen PD-L1. Remember, PD-L1 convinces attacking tumor-infiltrating lymphocytes, as depicted here, that the tumor isn't a tumor at all, but that it's normal tissue. It's telling this T cell, well, you may have gotten here, you may have detected that something's wrong, but there's really nothing wrong. Just move along. Leave me alone. The way anti-PD-L drugs work is that it blocks that PD-L1 tumor, not allowing it to tell the T cell to leave it alone, resulting in the T cell killing the cancer. Immune therapy has been a real game changer lately and has been very effective to treat patients who have highly immunogenic tumors such as melanoma, lung cancer, and kidney cancer, but has only shown minimal results for tumors that are not very immunogenic like breast cancer, pancreatic cancer, and colon and rectal tumors. We hope that we can treat minimally immunogenic tumors with focused ultrasound ablation and make them more immunogenic. For our first study of focused ultrasound ablation to treat breast cancer at the University of Virginia, we combined focused ultrasound ablation with a drug called pembrolizumab. Pembrolizumab is a programmed cell death 1 inhibitor and has been shown to be effective against treatment of some triple-negative breast cancers, which tend to be more immunogenic, but pembrolizumab is not effective against estrogen receptor positive tumors, which tend to fly under the radar of the patient's immune system. That brings us to our first study at the University of Virginia to treat breast cancer with focused ultrasound ablation, namely Breast 48, which utilized focused ultrasound ablation plus the drug I just mentioned, pembrolizumab, to treat patients with stage 4 breast cancer. The idea behind this is that we know that focused ultrasound ablation can cause an acute thermal injury to the tumor and initiate a brisk local immune response. As part of that immune response, CD8 positive T cells would then traffic to and infiltrate into the damaged area of the tumor, and then we could give pembrolizumab, which could upregulate that local cellular immune response, hopefully resulting in a brisk systemic response as well. The primary objectives of Breast 48 were to assess the adverse event profile of pembrolizumab and focused ultrasound ablation therapy in patients with metastatic stage 4 breast cancer, and to determine whether the addition of pembrolizumab to focused ultrasound ablation increases the proportion of CD8 positive tumor infiltrating lymphocytes into the primary and periablative zone. Stated more simply, the concept behind Breast 48 was as follows. We know that pembrolizumab is able to induce an immune response in some breast tumors, but not most. We also know that focused ultrasound ablation is able to induce an immune response in most tumors, but that local immune response is typically not enough on its own to be curative. And what we were testing was the idea that might the combination of focused ultrasound ablation and pembrolizumab be more effective. This is the schema for that trial, Breast 48. In the first arm, or arm A, patients got pembrolizumab after the focused ultrasound ablation, and in arm B, patients got the pembrolizumab prior to focused ultrasound ablation. So like I said, patients to be included in this trial had advanced or stage 4 breast cancer, as well as the other inclusion criteria on this slide. Here are ultrasound images from one of the first breast cancers that we treated. Here's the tumor within the breast. Here's a clip as seen on ultrasound within the cancer. Here is an image from the treatment plan, and as you can see, we delivered focused ultrasound ablation pulses to only within the tumor. We did not treat any of the surrounding tissue. 22 days after the ablation, we biopsied tissue in the periablation zone and tissue from within the ablated region of the tumor. This very busy slide shows a series of photomicrographs. In this column, this is before treatment within the tumor. This is at day 22 in the area of the tumor that we ablated, and this is at day 22 in the peri-ablative zone, in this case still within the tumor itself. They show an increase in the number of CD8 positive T cells that infiltrated into the tumor as compared to pretreatment, as well as an upregulation of PD-L1 within the tumor, again as compared to pre-treatment. Breast 48 is now completed and closed to accrual, and the final data from that study is currently being analyzed. However, the preliminary data was intriguing, and we've now come to the point where it's time to open a study looking at focused ultrasound ablation to treat patients with early stage breast cancer. Early analysis of the data from Breast 48 indicated that focused ultrasound ablation and pembrolizumab was able to change a patient's immune response to breast cancer. Preliminary results demonstrated an altered tumor microenvironment in which there was an increase in anti-tumor T lymphocytes, both CD4 and CD8 positive, trafficking to and intercalating into the tumor. These T lymphocytes are strongly associated typically with an increase in the immune response. However, we also found that shortly after upregulating that immune response with the T cells, myeloid-derived suppressor cells also traffic to the tumor. The problem with this is that myeloid-derived suppressor cells tend to tamp down an immune response. So we were quite disappointed in this because as soon as the T lymphocytes upregulated the immune response, the myeloid-derived suppressor cells tended to tamp it down. So the tumor was still able to work its magic at running below the radar of the immune system. In addition, mouse models in the Price lab from studies that were done concurrently with Breast 48 demonstrated similar findings. So after demonstrating that focused ultrasound ablation could initiate a localized immune response in patients with stage 4 breast cancer, we began developing a trial to treat patients with earlier stage breast cancer. That trial is Breast 54, and Patrick Dillon is the PI on this study. In this study, we use focused ultrasound ablation in combination with low-dose gemcitabine to augment the immune control of early stage breast cancer. So why use gemcitabine? Gemcitabine is a drug that is used to treat breast cancer, but its effect on the tumor is modest. However, in preclinical findings in the Price lab at UVA in mouse models, gemcitabine has been shown to decrease circulating myeloid-derived suppressor cells. And as you recall from my earlier comments on Breast 48, that myeloid-derived suppressor cells traffic to the tumor after we perform focused ultrasound ablation and tamp down that immune response. What we hope to do in Breast 54 is pre-treat patients with gemcitabine, drive down the myeloid-derived suppressor cells, and then initiate an immune response in the patient's tumor with focused ultrasound ablation. And as the myeloid-derived suppressor cells can't traffic to the tumor, we hope to initiate a much more robust and durable response. Here's one quick slide from Dr. Shabani's work in the Price lab, which demonstrated that focused ultrasound ablation plus gemcitabine was more effective than gemcitabine alone or focused ultrasound alone at controlling both the primary tumor and lung metastasis in their model. The study Breast 54 will have 3 arms. In arm A, patients will receive gemcitabine alone. In arm B, they will undergo focused ultrasound alone. And in arm C, they will receive a combination of gemcitabine and focused ultrasound, with the gemcitabine being given first. After those treatments, all the patients will have the standard of care therapy, which will include sentinel lymph node biopsy, definitive surgery, and will collect tissue from the tumor at various stages. After their surgical treatment, they'll receive standard adjuvant therapy and be followed for 5 years, and will collect data over the entire time period. So for clarity, in Breast 54, there'll be 3 arms. In arm A, patients will receive gemcitabine alone. In arm B, patients will undergo focused ultrasound ablation alone. And in arm C, they'll receive a combination of the two. Patients will be randomized 1:1:1 with a total of 48 subjects, and that means 16 patients in each arm. As Breast 54 is an exploratory study, we'll be looking at the primary endpoints of safety of focused ultrasound ablation, safety of gemcitabine alone, as well as the combination of the two. And we'll be looking at the following primary endpoints: adverse events, time to surgery, and positive margin rate. There are multiple secondary objectives and endpoints, but they are too numerous to review due to the time constraints of this presentation. However, they are presented on this slide as well as the next. Patients that will be included in this study have early stage breast cancer, in other words, clinical stage 1 to 3 breast cancer. Their tumor can be of any receptor status, and if genomic profiling is performed, they should be at high risk. However, genomic profiling is not required in order to be entered into the study. For safety in Breast 54, tumors must be at least 5 millimeters from the skin, and the rib to treatment target zone must measure greater than 1 centimeter. Again, we're only treating inside the tumor, and more specifically, the goal is not to treat the entire tumor, certainly no more than 50% of it. Here is a picture of the first specimen from a patient that we treated. This is after treatment, after the tumor has been excised, and you can see there is a necrotic portion of this tumor where we provided focused ultrasound ablation juxtaposed to viable tumor. And again, this is our intent. We're going to obtain biopsies for evaluation of the necrotic area and of this transition area, as well as of the viable tumor. Breast 54 is ongoing. It opened earlier this year. There are 3 patients who have been enrolled to date, and we soon hope to receive an updated device from Theracleon, which will allow us to treat patients with smaller tumors so that we can increase enrollment rapidly. So to wrap things up, at the University of Virginia, we have utilized a stepwise fashion to evaluate the use of focused ultrasound ablation to treat tumors in the breast. We started by treating benign tumors, namely fibroadenomas. We then moved on to treating patients with advanced stage 4 breast cancer with Breast 48, and now we're on to our study called Breast 54, in which we're evaluating the use of gemcitabine and focused ultrasound ablation to treat patients with early stage breast cancer. We strongly believe that focused ultrasound ablation has the potential to be both a local and systemic therapy, and we will be leveraging that concept in current trials at the University of Virginia, which use focused ultrasound ablation in combination with immune therapy for patients who otherwise would have a very low likelihood of response to immune therapy alone. Finally, I'd like to thank the following organizations and individuals without whom we couldn't make this happen. Thank you for your attention.
We gratefully appreciate the Focused Ultrasound Foundation for giving us the opportunity to present to you today on the topic of tempering the hostile tumor microenvironment to boost immunotherapy outcomes in triple-negative breast cancer. I will present the first half of this webinar, and my colleague, Dr. Mike Nemeth, will present the second half. We are faculty members in the Department of Immunology at the Roswell Park Comprehensive Cancer Center in New York. My research interests focus on immune suppression, myeloid tumor biology, and immunotherapy, whereas Mike's research interests reside in hematopoiesis and molecular biology of cancer. We believe such complementary expertise was important to move this project forward. And the work that we're presenting today has been provisionally accepted in the Journal of Clinical Investigation. At this time, we wanted to provide a brief background about our project. Metastatic triple-negative breast cancer, or TNBC, is an aggressive subtype with limited therapeutic options. Up to 20,000 new cases of metastatic TNBC are reported annually. Survival approaches 18 months post-diagnosis with conventional therapies. However, with the advent of immunotherapy, such as immune checkpoint inhibitors, or ICIs, survival in metastatic TNBC patients receiving chemotherapy plus an ICI agent has increased to approximately 25 months. While very promising, we believe there is still room to improve outcomes using ICIs, and we believe one key reason for limited efficacy to these inhibitors is immune suppression, which antagonize the benefits of ICI activity. Now, before we proceed to explain why immune suppression is critical to ICI response, we want to briefly review how these agents work. Populations of blood cells known as T lymphocytes are critical in host defense against disease, including cancer. These cells have the capacity to bind to and directly kill cancer cells, and this occurs through multiple steps simplified here as signal 1, which is recognition of the target cell by the T cell receptor, followed by signal 2, which is a co-stimulatory event important for expanding the T cell army. Once the target population is eliminated, the T cell response shuts down through a process involving immune checkpoint proteins. However, in cancer, this negative regulatory process, coined the brakes, is sustained, leading to T cell inactivation, also known as exhaustion. Two such immune checkpoint proteins are shown here, one called CTLA4 and the other PD-1. And blockade of these pathways using ICI agents removes the brakes on T cells, leading to sustained T cell activity across multiple cancer types. And for these discoveries, both Drs. Allison and Hanjo received the Nobel Prize in Physiology and Medicine in 2018. For the remainder of our talk, we will focus on ICI agents targeting the PD-1 pathway, which is now an FDA-approved intervention for late-stage TNBC when combined with standard of care therapies. Now let's return to the concept of immune suppression and why that is an important process to target to improve ICI activity. The tumor mass, also known as the tumor microenvironment, is complex and comprises diverse cell types, including T lymphocytes. One major infiltrating cell type includes the myeloid population, which are blood-derived like T lymphocytes. However, in the tumor microenvironment, these myeloid cells have been co-opted to aid or drive cancer growth through suppression of T cell activity, even in the face of these ICI agents. This final image combines everything to underscore the cellular complexity of the tumor microenvironment and the plethora of potential therapeutic avenues for intervention. One prominent myeloid cell type within the tumor microenvironment is the myeloid-derived suppressor cell, or also known as MDSCs. So MDSCs arise from the myeloid lineage in the bone marrow in response to tumor-derived factors secreting during the neoplastic process. Ordinarily, the myeloid lineage elicits various cell populations crucial for normal immunity, such as those shown here. Once generated, MDSCs inhibit the expansion and activity of T lymphocytes. They're found in patients with diverse cancer types, including breast, and notably, high blood MDSC percentages or frequencies have been correlated with poorer outcomes. So given the importance of MDSCs in cancer, it therefore makes sense to design strategies that minimize MDSC burden to improve response to these ICI agents. We know that MDSCs arise from the bone marrow during cancer growth and then travel to the tumor microenvironment where they exert their suppressive actions. Most strategies in the field attack MDSCs in the tumor microenvironment without considering their origin. Therefore, such strategies are unlikely to achieve durable control of MDSC numbers because MDSCs that are removed may be replenished by the bone marrow. Our concept is different. We developed a novel approach to block their production and to restore normal myeloid cell differentiation, thereby potentially barring MDSCs as a barrier to the activity of these ICI agents. Now I would like to turn over the podium to my colleague, Dr. Mike Nemeth, to present the second half of our presentation on how we apply this concept to bolster anti-PD-1 based ICI activity in preclinical models of TNBC.
So thank you, Scott. And to develop this approach, we looked to the scientific literature in the field of acute myeloid leukemia, or AML. This is a highly aggressive cancer of myeloid cells, and we therefore reasoned that therapies that targeted myeloid cancer cells could also be applied to MDSCs. In 2016, David Sykes from Harvard published a report that a drug that inhibits an enzyme called dihydroorotate dehydrogenase was effective at causing myeloid cancer cells to become normal myeloid cells. And so therefore we asked whether a drug that could cause myeloid cancer cells to become normal cells would also cause MDSCs to become normal myeloid cells. Importantly, in this paper, Dr. Sykes identified a particular drug called Brequinar, or BRQ. And we decided to test Brequinar in our studies based on prior work demonstrating that this drug could be safely given to patients and is currently in clinical trials in AML. And so we then tested whether Brequinar could combine with an immune checkpoint inhibitor, in this case anti-PD-1, to suppress tumor growth in multiple preclinical models of triple-negative breast cancer. And here we are showing the results from one model. We found that neither Brequinar nor anti-PD-1 alone had much effect on tumor growth. However, when the two agents were combined, we saw a significant reduction in tumor growth. Importantly, we also saw that the combination reduced the number of metastatic tumors. Interestingly, Brequinar alone also appeared to reduce the number of metastases, and we are currently performing additional studies to further explore this outcome. We then asked whether Brequinar was having an effect on the bone marrow in MDSC development, and we have done several studies to answer this question. And here I would like to highlight one particular finding. We've used a revolutionary new technology that allows us to identify genes that are turned on or off in individual bone marrow cells, thus allowing us to determine the expression pattern of genes for a particular cell. When we analyze these gene expression patterns from bone marrow cells isolated from our preclinical models, we found that for the population of cells that is highlighted in these plots as bright green, that Brequinar treatment restored the gene expression pattern of these cells to what we observed under tumor-free conditions. This is important because work from our lab and others have demonstrated that these bright green cells, in fact, represent the starting point for MDSC development. And in subsequent studies of these cells, we found that Brequinar treatment inhibited their ability to develop into MDSCs. So to summarize the key findings and conclusions from our collective studies. So first, we found that Brequinar plus anti-PD-1 had robust anti-tumor activity, both as primer inhibiting primary growth, as well as the development of metastases. Second, we found that Brequinar suppresses MDSC function, and that the effectiveness of this combination was dependent on the reduction of MDSC burden. We have not shown these results today in this webinar, but would be happy to discuss them further. And they, in fact, are a critical part of that manuscript that has been provisionally accepted at the Journal of Clinical Investigation. Third, we found that Brequinar inhibited the ability of bone marrow cells to develop into MDSCs, and potentially by changing their gene expression pattern. And this is work that is currently being done by our labs. And so these findings are summarized in this model, in which the ability of the tumor to cause the development of MDSCs in the bone marrow is suppressed by Brequinar. This results in the development of normal myeloid cells, thus relieving immune suppression in the tumor microenvironment and allowing activation of an anti-tumor immune response with checkpoint inhibitors. We believe that the significance of our research is that it will impact clinical practice by improving outcomes for patients with metastatic triple-negative breast cancer who are unresponsive to standard of care immunotherapy. Further, our data will be useful to develop clinical trials that will test the efficacy of targeting MDSC development in patients with other types of breast cancer and in different clinical contexts, and perhaps even cancer types beyond breast cancer, where MDSCs are a barrier to effective therapy. We would like to thank our funding partners as well as the Breast Cancer Alliance community for their support of this early stage research. This work was done as a collaboration between the Abrams and the Nemeth labs, and especially we would like to highlight Sean Colligan, who has just defended his PhD thesis largely based on this research. And we also thank our colleagues both at Roswell Park and outside institutions for their collaborative efforts, especially Annette Colden, a patient advocate at Roswell Park, who has helped us develop our research to best serve the community. And finally, if you would like to discuss our work further, please feel free to get in touch with us. Thank you, and we will be happy to answer any questions.
Wow, that was incredible. Thank you, Doctors Brenin, Nemeth, and Abrams, for sharing your research and your thoughts and for explaining it in such an easily understandable format. Really amazing stuff. And it gives hope to soon improve the care for many patients that are enduring other treatments for breast cancer. So thank you for sharing that. I'd like to now introduce Dr. Natasha Shevani, who's going to lead our discussion. I think there's so much to talk about, Natasha. I mean, really promising research.
I couldn't agree more, Susie. And I just want to say thank you to all of our speakers. I think both talks were extremely compelling, and there's this common ground around inciting immune responses, but also curbing the immunosuppression in breast cancers. And I did take note, maybe for an opening question, of a statement that Dr. Brenin made. As we think about the power of immunotherapy, I think something we think a lot about is inciting what we call an abscopal effect. And this is something that Dr. Brenin, myself, and colleagues have talked quite a lot about. Dr. Brenin, you said that you believe FUS has the potential to be both a local and systemic therapy. I was wondering if you could talk to us a little bit about the abscopal effect and what your vision and thoughts might be around how we can achieve that effect with focused ultrasound.
Sure. Thank you, Natasha, and also, thank you for your pioneering work in the Price Lab at UVA, which really has provided the basis for our current study, Breast 54. So definitely indebted to you, as well as the rest of the Price Lab and our immune therapy program here. So in terms of the abscopal effect, I think the first thing to understand is that I'm a breast cancer surgeon, and we have great ways to treat breast cancer. A lumpectomy is a very well-tolerated procedure, typically results in excellent cosmesis, and is curative for the vast majority of patients that we operate on. So any new treatments for breast cancer really are going to need to be held up to that bar. But other than the slide that I showed, that scar versus no scar, there's probably something that I believe is even more important, and that's the idea that we may be able, with focused ultrasound ablation, to not only treat the primary tumor, but have an impact on metastatic sites, because I can remove the primary tumor. I can cure the patient of her primary tumor almost all the time. But what I cannot do is to adequately treat in any surgical way systemic disease. And what I mean by that, we do have a very diverse audience. So I'm just going to bring this down to a level that everybody can understand. When we say systemic disease, we mean individual cells or clusters of cells that have left the tumor or metastasized to elsewhere in the body. When we cure someone of cancer, we have to not only remove the primary tumor, in the case of breast cancer, the tumor in the breast, or maybe also tumor that is spread to lymph nodes, but we also need to make sure that there's no metastatic disease, which is usually subclinical or unknown at the time of diagnosis. With focused ultrasound ablation, we may be able to treat the primary tumor, incite the immune response that we've all been talking about in this presentation, and allow the patient's immune system to begin to identify the breast cancer as something that shouldn't be there and attack the breast cancer, not only at the site of the primary in the breast, but then those activated immune cells circulate throughout the body, finding small metastatic deposits and attack the tumor there as well. Hopefully curing the patient of the cancer and improving prognosis even more. So in a nutshell, that's what I believe is the potential benefit of moving towards an ablative treatment with focused ultrasound ablation, and then ramping up that immune response in the way that these presentations have discussed for patients with breast cancer as compared to surgery alone.
Thank you so much. And as you know, this is something that we both and with the foundation as well care mutually about enabling with focused ultrasound. And I just want to emphasize to the audience that one of the exciting things about meeting that goal is we'd be doing so with a technology that is completely non-invasive and non-ionizing. So from the standpoint of patients' comfort and off-target toxicities, there are some really great benefits there too. Doctors Abrams and Nemeth, I am so compelled and excited by the work that you are doing with Brequinar. And I do want to maintain this theme of abscopal effect by returning to the fascinating observations that you all made in the lung mets in your preclinical models. So I'm wondering if you can speak to us a little bit about whether you've dug deeper and implicated any type of abscopal response that may be driving that. And secondarily, maybe you could tell us a little bit more about your vision for translating this work, if the gears are already in motion to take this combination of anti-PD-1 and Brequinar into the clinic. So a two-part question for you.
Okay. Yeah, I'm sitting next to my colleague here, so I'll take the first part.
Sure.
Yeah, so that was a really intriguing finding that Brequinar, even as a single agent, had an anti-metastatic effect. We do know with the combination it's even more effective, and we were able to demonstrate that in multiple preclinical models now. We believe it all comes down to the nature and the dynamics of distinct tumor microenvironments. So we believe that the mammary or breast tumor microenvironment is fundamentally distinct from that of the lung microenvironment. Now we're beginning to try to unravel what those differences are, and it may have something to do with the types of T cells that infiltrate the tumor, or there could be differences in the nature of the MDSC populations or other myeloid populations within the lung that may be more susceptible to treatment with Brequinar. So we're going to be exploring different possibilities that it could explain why, in that case, single agent therapy is effective. But I do think more of it will come down to the T cell response, and that the T cells that infiltrate the primary tumor may express a certain type of functional profile, whereas the T cells that infiltrate the lung microenvironment may express a more productive or anti-tumor profile. And those are the kinds of things we're going to be looking at. We're going to focus on what we call T cell repertoire diversity and clonality. And I do think that would make a very testable hypothesis, partly because we had additional data that, because of time, we weren't able to share with you, but the T cells that do infiltrate, at least in the microenvironment of the mammary gland, show an activation phenotype consistent with strong or robust anti-tumor characteristics. So that's why we want to use that information as a foundation to explore differences in the T cell response. But again, it could also deal with susceptibility of the different myeloid populations in one microenvironment versus the other. I'll have my colleague Mike answer the second part.
Right. So one of the reasons why we eventually selected Brequinar is because there actually is a long clinical history of using these drugs and similar drugs in that class of these dihydroorotate dehydrogenase inhibitors. They've been found to be safe. Interestingly, they've been found to be fairly ineffective as monotherapy in cancer. And that's actually a finding that we replicated in our preclinical models. But our hypothesis is that you need to combine those agents with an immunotherapy to get the true beneficial effect. And that is something that and because though there is such a robust clinical history of the use of these compounds in several different types of cancer, none of which had led to a toxicity that would preclude their use, we think we can rapidly translate our preclinical data into a clinical trial, combining Brequinar or a similar agent with standard of care immunotherapy in these patients.
Wonderful. Well, thank you so much both for those enlightening answers. And as we are entertaining questions from the audience, we just had one come through that I'd like to read. And I believe this is again directed to you both, Dr. Abrams and Dr. Nemeth. Actually relates to the conversation we were just having about the metastases. And I have to say, I'm particularly interested in the answer to this question because we are within the focused ultrasound community also very interested in how focused ultrasound can not only curbs metastasis, but how that is reflected within the blood. And as you know, there are circulating tumor cells within the blood, right? So this concept of liquid biopsy is a resonant theme in the field. And one of our audience members asked, is the reduced number of lung metastases due to reduced engraftment in the lung tissue of circulating tumor cells? Or is it actually just due to a reduction in the growth of metastatic foci? So have you all explored CTCs at all in your preclinical studies?
No, we have not. It's an excellent question. And I do think at this point, both options are on the table. I do think it probably has more to do with colonization in the lung. We've done some early experiments in different settings where tumors can arrive to the lung. And I think it's at that point, once they arrive to the lung, so there's no difference in trafficking to the metastatic site or seeding within the metastatic site. It's more about survival and expansion within the metastatic site. So the first hypothesis that we're going to test is whether or not it affects survival and expansion. So once the cells do arrive to the lung, then we think that the immune response, which appears in the lung, and again, to Dr. Brennan's point, it travels systemically. It follows the path to the lung where the tumors reside, and it's within that microenvironment where the immune response would then take place. And we think it has something to do with the robust anti-tumor activity that then is expressed within that microenvironment. But it's also possible that it could affect invasion and metastasis. So we're going to do experiments where we can bypass the confounding effects of the primary tumor by doing what's called experimental metastasis models, where you can directly analyze the cells within a metastatic site by injecting cells IV, or we can do what's called surgical resection of the primary tumor and then evaluate the multi-step process by which this therapy works in impeding metastasis, whether it's migration to the tumor site in the lung or by affecting colonization and growth. But right now, we're leaning more towards the hypothesis that it affects the survival, growth, and expansion within that metastatic site.
Wonderful. Thank you. I do have another question for Dr. Brenin. Dr. Brenin, I think it's really impressive the scope of the trials that you discussed today, because there is at least one trial that's evaluating focused ultrasound and immunotherapy in the context of metastatic breast cancer, but then the recent FUS plus GEM trial that's opened is more focused on early stage breast cancer patients. And across these trials, there are also a variety of different hormone receptor statuses that are represented. And I think all of this speaks to an underlying heterogeneity that, we as preclinical researchers are very curious about, because it would be really interesting to know where and when focused ultrasound could plug in as relates to the various stages of breast cancer. So I'm wondering if you can comment on how you envision focused ultrasound, dare I say, becoming a mainstay of breast cancer treatment, for metastatic versus early stage disease. How do you envision FUS plugging into the clinical paradigm?
Well, obviously, that's going to depend on how these studies go.
That's right.
So,
a loaded question.
Yeah. So when we think about the treatment of breast cancer over time, and you go back, I mean, I wasn't alive more than 58 years ago. But you know, in the late 1800s and before that, the surgical treatment and the entire treatment of breast cancer really revolved initially around less radical surgeries, and then once we learned how to do more radical surgeries more safely, a fairly radical surgery, a radical mastectomy where we remove the entire breast, all of the skin of the breast, the nipple, the pectoralis major muscle, the pectoralis minor muscle, and all of the lymph nodes underneath the arm, so much so that there was not enough skin typically left to close, so a skin graft. And even before the skin grafting, just a blood clot was left on the chest wall. You know, that's obviously how it started. And then over time, we realized we probably didn't need to be quite as radical. And we started doing mastectomies, but removing less tissue. And then we realized we probably didn't need to remove the entire breast to treat breast cancer. We could just remove the tumor from the breast, but still do a large operation underneath the arm called an axillary dissection that also had a lot of morbidity to it, or in other words, side effects, arm swelling being the primary one. And then really in the late 90s, we realized, well, maybe we don't need to remove all of those lymph nodes. We can even scale the surgical treatment back a little bit further. And we came up with something called sentinel lymph node biopsy, where we removed just a few lymph nodes, but then went back to remove all the lymph nodes if there was any suggestion that there might be cancer that is in those lymph nodes. And then in the mid-2000s, you know, like about 12 years ago, we realized, well, we don't even have to remove lymph nodes, even if there's cancer in them. We could treat those lymph nodes just with radiation. So we've just scaled back our treatments for breast cancer from very radical and invasive to less invasive with each evolution in our treatments. More recently, we're probably coming to the point where we don't even need to remove lymph nodes anymore. And, you know, the surgery is getting less and less invasive. And I think that's where the ablative therapies are going to fit in. And that's the next logical step, to maybe be able to forego surgery entirely and treat tumors with an ablative method. There are various ablative methods that are out there. Cryotherapy probably is the most prominent one currently in the treatment of breast cancer. And I believe that focused ultrasound ablation, though, as compared to cryotherapy, has advantages because of the higher precision that we can deliver the ablative therapy to the tumor for our patients compared to cryoablation. And I also believe, all right, my hope is, and certainly our preclinical data would support the idea that we're going to do more than just get rid of that primary tumor, but that we also may help patients fight that cancer on a systemic level and increase cure rates. So not only can we be less aggressive in our treatments, less invasive in our treatments, actually treat the cancer without surgery, but also do it better by increasing the likelihood that we're going to cure the patient of her disease. So that's really what I hope will happen. The timing of that, you know, change in the treatment of cancer in general, and certainly in breast cancer, takes time. We need to do these studies in a stepwise fashion in the way that I laid out. You know, if we're successful, we could hope that over time we could do larger studies, prove efficacy. And then it's a pretty easy decision at the end of the day for patients, right? If we have a treatment that's not invasive and is actually as good or perhaps superior to surgery, very few patients are going to line up to have an operation when they don't have to have it. You know, it's really that simple. You know, when I present these this information and studies to breast surgeons, I'm a breast surgeon, right? I get up at big meetings, the American Society of Breast Surgeons and the Society of Surgical Oncology, and I'm starting to talk about, hey, we don't need to do surgery. You know, people are kind of like, what are you talking about? You know, that's nuts. Lumpectomy is not a big operation. People do really well with that. But I point out at the end of the day, even if you actually take a step back, and even as a surgeon, you know, if I offered you the opportunity tomorrow, hey, do you want to have an operation tomorrow, or you want to not have an operation tomorrow? Very few people are going to say, yeah, let's go get an operation. So that's how I think this is going to move forward.
Well, thank you so much for those thoughts, Dr. Brenin. I just want to say, I think it's especially exciting and encouraging to hear from a breast surgeon that you subscribe to this idea that focused ultrasound can play such a profound role in potentially even, replacing the need for surgery one day. So we are right up on
I'm going to deny that statement if it becomes more public. But yes, I agree.
But I think from all of the speakers that we heard from, these are really exciting and promising strategies. I want to thank everyone again for educating us today and for your time. And I will turn it back over to Dr. LeBlanc.
Thank you, Dr. Shabani, for leading such a great discussion. And, this conversation has been so interesting with, manipulating the tumor microenvironment to make our immunotherapies and our own immune system work better. And I'm so excited about it. And more importantly, working with disease-specific organizations like Breast Cancer Alliance results in forming these types of collaborations and sharing of information that can hopefully more quickly lead to clinical trials and more effective therapies for patients. So I'm so excited about all of us getting together to discuss this. Yanni, did you want to say a few words as well?
I'm just always so impressed and humbled, truthfully, by these presentations. And it's a feather in BCA's cap to be supporting Dr. Abrams and Dr. Nemeth. And I think there's so many misconceptions about breast cancer, one of which is that breast cancer is a singular disease. And those of us who work in different aspects of this field understand that it's going to take something different for each type. Everybody's pathology is different, everyone's reaction is different. And so to have brilliant doctors and scientists on the front lines trying to figure out how to treat everyone where they are in terms of what they need, particularly with something like triple negative, which is so elusive. I just think it's really exciting, and we're thrilled to be a part of moving that needle forward in any way we can. So thank you for allowing us to be here today and to continue to collaborate with you.
Well, this concludes today's webinar. I want to thank Dr. Abrams, Dr. Nemeth, Dr. Brenin, and Dr. Shabani for participating, and Breast Cancer Alliance for your partnership. If your questions were not answered or you want additional information, in addition to finding out about clinical studies, visit our websites at busfoundation.org or breastcanceralliance.org. Importantly, a recording of this webinar is going to be available on both of our websites, hopefully within the next 24 hours. So thank you for joining us today and for sharing this cutting edge research. It wouldn't be possible without all of your contributions. Please stay tuned and go to our websites for future newsletters and events to continue these conversations. So thank you, everybody, and I hope you have a great day.